Let's now begin by introducing our speaker from TriLink, Jordana M. Henderson, PhD. She's a scientist with TriLink. Thank you for joining us today, Jordana. Now I'll hand it over to you to begin the presentation.
Thank you, Michael, and thank you all for having me and tuning in today. I'm Jordana Henderson, master's degree. Thank you for the upgrade, though. I'm a research and development scientist at TriLink Biotechnology, and I'm honored and excited to share with you today about streamlined mRNA synthesis using CleanCap technology. A quick overview. I will be breaking the session into two sections. First, we will review mRNA manufacturing processes for vaccines and other therapeutics, dive into an understanding of the molecular structure of mRNA with an emphasis on the 5' cap, what it is, why it's important, and how it affects the manufacturing workflow. To end the first section, I will share data with you from an in vivo case study using mice to compare the efficacy of the various 5' cap options using mRNA LNP delivery systems. In the second half of the talk, we'll take a virtual step back into the research and process development labs to explore how we can further improve our manufacturing process in terms of in vitro transcription, as well as mRNA purification optimization. To end, I will share how these manufacturing improvements translate to in vivo expression with additional data from our case study. As we have all learned from the past year, mRNA vaccines are a quick and efficient option, especially in the face of global viral pandemics. Making the mRNA vaccines is typically done in two parallel manufacturing workflows to create the nucleic acid of choice that can be mixed with solubilized lipids to form lipid nanoparticles, or LNP. The LNP serves to both protect the mRNA from outside degradation sources, as well as creates the delivery vehicle for the message into a cell. At TriLink, we specialize in the top pathway, starting with our newest offering, plasmid DNA production, which is taken all the way through to linearization, transcription, and purification of mRNA for your vaccine and other therapeutic needs. So before we go any further, what is an mRNA and why do we choose to use it? At the most basic level, messenger RNA is the transient message between your permanent genetic DNA code and the dynamically expressed proteins within your cell. As a therapeutic option, mRNA offers certain advantages over DNA or proteins. For certain proteins, they are highly modified post-translationally, which can be difficult to replicate in a lab, especially in a quick and cheap manner. Conversely, mRNA has an advantage over DNA-based therapeutics with a reduced safety risk of genomic insertion because mRNA is a self-destructing message. The process to manufacture mRNA in a lab is called in vitro transcription. This enzymatic reaction mimics the same process that occurs in our cells every day. With the appropriate reagents, any message can be produced in a tube. And requirements include a DNA sequence to act as a template for the message of choice. This DNA template is recognized by a DNA-dependent RNA polymerase through a specific promoter sequence. Next, we add our ribonucleotides, or NTPs, to serve as the building blocks of the message, which will incorporate from 5' to 3' in a template-dependent fashion by the RNA polymerase. It is common to use bacteriophage-isolated RNA polymerases, such as a T7 RNA polymerase, as they do not require additional proteins to function. Simply add magnesium in a buffered solution for your in vitro reaction. However, in most reactions, there is an inherent and aberrant activity of these polymerase enzymes that results in double-stranded RNA, an undesired byproduct of the IVT. Optionally, a cap or cap analog can be included in the reaction to synthesize fully mature mRNA molecules in a single step. On the screen now is the structure of a mature single-stranded mRNA, the desired molecule for exogenous therapeutic function. From left to right, there is a 5' cap, 5' untranslated region, or UTR, followed by an open reading frame that codes for your message of choice. Typically, this reading frame only contains the exons such that no in vitro or in vivo splicing is required. The ability to replace this part of the sequence by simple DNA cloning techniques for your DNA template is what makes mRNA such a versatile and universal platform. The longer the message, however, the more fragile it becomes, as the risk of any cleavage to your phosphate bonds can increase. Downstream of our reading frame is a 3' UTR, or untranslated region, and finally a polyadenosine, or poly-A tail. Each of these components of the mature mRNA molecule is vital for translation of your message. All components except for the 5' cap can be encoded by your DNA template design. Which brings us to our first focus topic of the day, the 5' cap. When I say a 5' cap in today's talk, we're referring to the canonical inverted N7-methyl-G shown at the very left in the cap0 structure. This base is connected to the rest of your RNA sequence by a 5' triphosphate bridge. Within this canonical cap exist various modifications conferring a cap1 or cap2 not shown structure. On the left, cap0 contains no additional modifications and is not found in higher eukaryotic mRNAs. In other words, it is most commonly found in viruses. In contrast, on the right, methylation of the 2' position of the ribose sugar of the first base in an mRNA creates a cap1 structure. This cap1 structure is found among higher eukaryotic organisms. The cap structure serves several important functions in a cell, including nuclear export, splicing, translational initiation, protection from decapping enzymes and subsequent degradation, as well as self versus non-self recognition. In the context of an exogenously delivered mRNA to the cytoplasm for therapeutic purposes, we are not as concerned with the export from the nucleus or splicing. We are, however, concerned about the delivery of the exogenous nucleic acid and how it will be recognized and trigger alarms in the innate immune system. Toll-like receptors will recognize both single and double-stranded RNA in an endosome, the most common LNP delivery mechanism. And cytosolic receptors recognize 5'N in addition to double-stranded RNA contaminants. Improperly capped mRNAs, such as a cap0 or uncapped molecules with 5' triphosphates, are bound by IVT or RIG-I, respectively, while double-stranded RNA can bind PKR and or MDA5 pattern recognition receptors, triggering additional signaling pathways that ultimately result in the release of cytokines as part of an immune response to your foreign entity. It is important to note that a cap0 molecule bound to IVT may also result in the sequestration of that mRNA away from other cap binding proteins involved in translational initiation. Overall, this could result in less expression of the desired protein. As we have learned, the 5' cap is a necessary component of the messenger RNA, but as previously mentioned, it is not coded for by the DNA template used in vitro transcription. Thus, alternative strategies must be used in the manufacturing process to add a 5' cap to the mRNA. This can be done by two main strategies: enzymatic capping or co-transcriptional capping by a cap analog small molecule. Relative to enzymatic capping, co-transcriptional capping reduces the overall manufacturing cost of an mRNA and is done in fewer steps, ultimately saving you time and money. Cap analog technology has evolved with time, with each iteration improving on its predecessor. First generation cap analogs are GG dimers that result in a capped zero structure. However, there was no control in the orientation of the N7 methyl G relative to the unmodified G at transcriptional initiation. As the name suggests, second generation analog anti-reverse cap analog, or ARCA for short, resolves the orientation issue, but still only offers a cap zero structure. The newest generation, an invention by TriLink called CleanCap, allows for a Cap1 co-transcriptional IVT reaction. It is important to understand the different mechanisms between these two capping strategies so that we can analyze and see the benefits of co-transcriptional capping. On the screen now is a schematic representation of the in vitro enzymatic capping process, where a 5' triphosphate is first dephosphorylated to allow for guanyl transfer, followed by methylation at the N7 position to result in a cap zero structure. This concerted action can all be done by a single vaccinia capping enzyme. However, in order to convert the cap zero to a Cap1, a second enzyme, or 2'-O-methyltransferase, is required. For best results, we recommend a gentle heat denaturation of the RNA's complex secondary structure to ensure the 5' end of the molecule is accessible by both enzymes for this enzymatic capping process. Heating of the RNA in the presence of magnesium will result in phosphodiester bond cleavage, and therefore mRNA products from an IVT must first be purified prior to enzymatic capping. This technique and approach may be more difficult to apply to long mRNA sequences, as they are more easily prone to degradation. In contrast, co-transcriptional capping with a cap analog allows for a one-pot reaction. Among the cap analogs now available, we can see differences in their structure. On the left shows an ARCA GG small molecule. The anti-reverse mechanism stems from the 3'-O-methyl moiety on the inverted N7G, which serves to block polymerization from the T7 RNA polymerase. Of note, a 2'-O-methylation can also inhibit enzymatic polymerization. In an IVT reaction, G2P is the preferred ribonucleotide for initiation downstream of the T7 promoter sequence. When ARCA is present in the reaction, it can serve to replace G2P at initiation, resulting in a capped mRNA. Unfortunately, in practice, G2P and ARCA will compete for this initiation, and overall capping efficiency among the molecules produced in each batch can be quite low. To overcome this, G2P concentration is limited in the IVT reaction, while an excess of ARCA is used to improve cap analog initiation. This tweak in the reaction to limit one of the essential building blocks of the message also results in lower IVT reaction yields. On the right side of the screen, I'm showing TriLink's first CleanCap analog, or CleanCap AG, resulting in a Cap1 structure. CleanCap is a G trimer with 2'-O-methylation on the ribose of what becomes base 1 in the mRNA. With this analog, the start of the 5' UTR in the DNA template is changed from a G to an A directly downstream of the TATA box of the promoter sequence. The CleanCap analog can now serve as a dinucleotide primer and hybridizes to the DNA template for dimer initiation by the T7 RNA polymerase. Moving beyond the mechanism of each cap analog, let's next compare the results from a typical co-transcriptional cap IVT reaction. On the screen are representative traces from an LC-MS assay used to calculate capping efficiency. In this assay, the capping efficiency is determined as the sum of the uncapped masses over the, sum of the capped masses over the sum of all sequence-specific masses to provide a relative percent capped and percent uncapped. On the left, an ARCA IVT results in 80% of the mRNA molecules initiated by the cap analog at best, leaving 20% of your molecules uncapped. In contrast, CleanCap IVT results in 95% capping efficiency or higher. To review, each capping strategy has an impact on the mRNA manufacturing process. Enzymatic capping requires 2 additional enzymes to achieve a Cap1 structure, and it also requires an intermediate purification step to remove magnesium and other IVT components prior to the heat denaturation of the mRNA structure for best enzymatic capping results. In the middle, an ARCA one-pot transcription allows for a Cap0 mRNA, but at moderate to low capping efficiency. As such, this means that 20% or more of your mRNA molecules contain a triphosphate on the 5' end, which may trigger extra immune responses. For certain applications, it may be desirable to treat the mRNA with a phosphatase enzyme to convert uncapped ends to the 5' hydroxyl structures. And on the right, CleanCap IVT allows for a Cap1 mRNA in the most streamlined workflow, starting from a one-pot co-transcriptional IVT reaction to a single purification without the need for any additional processing steps. Overall, this allows for an easier scale-up of your manufacturing. Next, let's compare the outcomes of each capping strategy. I've summarized some of the key quality attributes to consider when deciding between these 3 capping strategies. In terms of IVT yield, given in mg of mRNA per mil of reaction, there is a significant benefit in choosing either the enzymatic capping or CleanCap, as the ARCA IVT results in the lowest IVT yield, on average only 1 to 2 mg per mil. In terms of process steps, or overall speed and turnaround time of your mRNA production, CleanCap has the clear advantage of utilizing the most streamlined manufacturing workflow, comparing 5 versus 7 total unit of operation. The quality of the final mRNA product by an average mRNA integrity, there is a slight disadvantage to using the ARCA, especially in the 7-step workflow incorporating a phosphatase step. Highest integrity is desired as it correlates to the mRNA efficacy. As previously reviewed, high capping efficiency is correlated with high mRNA expression. Cap efficiency is lowest in the ARCA IVT reaction, ranging from 60 to 80% Cap0 molecules on average, whereas enzymatic capping or CleanCap IVT results in the highest capping efficiencies at 95 to 99% Cap1 mRNA molecules. And finally, when deciding on the strategy for you, CleanCap is the most cost-effective mRNA manufacturing process. For the purpose of today's talk, as we move into data from our case study in the context of a streamlined manufacturing workflow, I want to focus on the 2 co-transcriptional capping strategies as they allow for the fastest and cheapest mRNA protection. This is a goal that we have seen in high demand over the past year in response to COVID-19 pandemic, and likely a demand that will persist in years to come for other pharmaceutical needs. Now ready to move into our case study. I apologize. Looks like we'll start on this slide. Okay, so the case study design is to compare luciferase mRNA expression in mice using different 5' cap analogs. All mRNA was synthesized at TriLink, sent to a first CRO for formulation into LNPs, and finally sent to a second CRO for injection into mice. I'll start by reviewing the IVT specifications and results of the synthesis at TriLink. In this study, we transcribed firefly luciferase, or FLuc for short, using N1-methyl pseudo-U modified base. FLuc was transcribed either with ARCA or CleanCap analogs, where the ARCA IVT is set up with a limited GTP concentration. Each mRNA was purified, and half of the material was subject to an extra phosphatase treatment and second purification step. This was done to investigate the effects of residual uncapped 5' triphosphates on mRNA expression in mice. Looking at the data, our first measurement compares mRNA yield. CleanCap significantly improves IVT yields with 4.7 mg per mil relative to ARCA cap analog at 1.3 mg per mil. The mRNA integrity was measured by IPRT HPLC UV detection, and the area under the curve of the full-length product was used to determine a percent purity. Results indicate that without phosphatase treatment, CleanCap mRNA has a higher integrity than ARCA mRNA. Similarly, with phosphatase treatment, CleanCap mRNA is better than ARCA mRNA, but overall, phosphatase reactions lower the quality of each final mRNA product. Results from our LCMS capping assay show significantly higher capping efficiency with CleanCap at 98% capped relative to ARCA at 80% capped. If you recall, the double-stranded RNA is an undesired byproduct contaminant from the T7 transcription reaction. As such, less double-stranded RNA, as measured by a J2 immunoglot, can be interpreted as the preferred and better outcome. J2 assay results shown here indicate that CleanCap mRNA contains less relative double-stranded RNA than an ARCA sample. And finally, two other key mRNA quality attributes were measured, looking at the clearance of our starting materials, including starting DNA template and clearance of all enzymes used to transcribe and quench the synthesis reaction. No significant changes were observed in our qPCR assessment of residual DNA, nor our nano-orange measurement of residual protein. Of note, all results were below assay limits for the residual protein detection. In summary, CleanCap improves 4 of the major mRNA manufacturing attributes when compared to ARCA. Next in our study design, the mRNAs were formulated into LNPs and injected into mice for comparison of cap analog function in vivo. The mouse study began with randomization of female CD1 mice into cohorts of 5 animals. Control group was injected with PBS buffer, and test groups received a single tail vein injection of 1 mg/kg mRNA LNP. Mice were weighed once a day to ensure no significant weight loss occurred as a result of the test samples. I'm happy to report that no such adverse events of significant weight loss or animal death occurred during this study. For the purposes of tracking mRNA expression, animals were also injected with luciferin substrate, and whole body luminescent images were taken at 7 time points throughout the study as indicated by the camera icons. As a way of measuring immune response to each test sample, blood samples were taken at 3 and 24 hours post-injection and analyzed for various cytokines. I will start by sharing the IVIS images of the luminescent mice. On the screen now, I am showing one representative animal from each cohort of 5 across all 7 imaging time points, starting with 3 hours on the left to 96 hours post-mRNA delivery on the right. On the top, we have our representative control animal where no luminescent signal is seen. In the middle, we have our representative CleanCap F-Luke mRNA animal with and without phosphatase treatment from left to right. In the bottom panel, we see the representative ARCA F-Luke animal with and without phosphatase treatment. As you can see by eye, the signal intensity of the CleanCap images is higher than the ARCA images, indicating more mRNA expression using CleanCap. When plotting the data from all 5 animals per cohort across the 7 time points, we see that the 2 blue lines represent in vivo expression by CleanCap mRNA, and the 2 green lines represent in vivo expression over time by ARCA mRNA. As you can see, at the 6-hour time point post-mRNA delivery, we observed the most luminescent signal in our CleanCap mRNA expression. This was 2.4-fold increase over the ARCA sample at the same time point alone. We next integrated the area under each curve and performed statistical analysis. In this graph, showing the average integrated flux or photons per second signal with standard error from all 5 animals across all 4 test samples, I'd like to first point out that no significant difference in expression was observed between the matched plus and minus phosphatase treatments. This was also indicated on the previous slide. We did, however, observe a significant increase in mRNA expression by students' t-test when using CleanCap mRNA relative to our Cap0 ARCA mRNA. And finally, we compared the cytokine levels from the blood draws as a means of comparing overall immune response to the different 5' caps of the otherwise identical mRNA molecules. In general, we observed that cytokine levels in response to CleanCap, as shown in blue, are lower than those in response to ARCA, shown in turquoise. To summarize, our in vivo data showed that all LNP mRNA delivery resulted in a mild immune response. This is to be expected when delivering any foreign molecule into a cell. However, no test animals suffered severe immune response, as indicated by the 100% survival rate and body weight maintenance of the mice. CleanCap samples elicit a lower cytokine response and higher protein expression than ARCA samples. I'd like to correlate these in vivo results to our in vitro results and remind you of the differences between the CleanCap and ARCA mRNAs. One utilizes a Cap1 structure with high capping efficiency and relatively low contaminating double-stranded RNA, while the other uses a Cap0 structure at lower capping efficiency with higher levels of relative undesired byproduct. By now, I hope I have convinced you of the benefits of using CleanCap co-transcriptional capping strategy for your mRNA production. CleanCap can streamline the manufacturing process, reducing the overall production costs and turnaround time. Relative to other cap analog strategies, CleanCap improves your mRNA as seen both by in vitro and in vivo metrics. CleanCap reduces the in vivo immune response relative to ARCA when tested in mice, and CleanCap also significantly improves the mRNA expression in vivo. This is the ultimate goal of your mRNA therapeutic. In our mouse study, we found phosphatase treatment did not impact the levels of luciferase expression or immune responses. At this point, I'd like to take a step back and ask how can we further improve our manufacturing processes, and how do these improvements affect the functionality of your mRNA? The goal here is to continue improving our processes, resulting in a robust and scalable manufacturing of high-quality mRNA with high function. We've already addressed ways to do this at the 5' cap, and we'll now take a look at ways to improve IVT yield, increase mRNA integrity or full-length products, decrease double-stranded RNA byproducts, all while maintaining a streamlined process. To increase IVT reaction yields, we tested several factors by design of experiment, or DOE, and used JUMP software to model these reactions. Test factors included your template concentration, T7 RNA polymerase concentration, NTP, magnesium, and CleanCap analog concentrations, as well as reaction time, reaction temperature, and reaction pH. The main goal was to design for a higher mRNA yield. However, we also measured mRNA quality, capping efficiency, and double-stranded RNA to ensure that new IVT conditions were not at the detriment of other quality attributes in our final mRNA. All of this was done across various sequences and lengths with the use of wild-type and modified NTP bases. On the right is an example of one of our IVT models, allowing for predictable outcomes in the event that one of your variables needs to be changed. I want to point out that these changes to the reaction did not require an increase in DNA template concentration, as this may not always be amenable to the same streamlined process we currently have in place. Production cost and time of your DNA template before IVT will also factor in to your final mRNA turnaround time. To summarize the results of these IVT optimization experiments, we can compare the yield, quality, capping efficiency, double-stranded RNA, and residual DNA and protein across 4 catalog sequences, ranging in length from 1 to 4.5 kilobases. In blue, our current IVT protocol results in 5 mg per mil with capping efficiencies by CleanCap Cap1 analog at 95 to 99% efficiency. The optimized reactions, shown in purple, result in higher mRNA yields with lower double-stranded RNA contaminant for an overall cleaner transcript. I'm excited to introduce to you today for the first time our new process, CleanScript IVT. When I talk about decreased double-stranded RNA, I wanted to show you some of that data. On the screen now, we see an anti-double-stranded RNA J2 immunoblot used to compare relative levels of this double-stranded RNA contaminant between equal amounts of mRNA. In the chemiluminescent image on the right, we start with a fully double-stranded RNA ligand to create a standard reference curve by dilution from 20 down to 1 nanogram. We then immobilize equal amounts, in this case 2 micrograms of test sample mRNA onto our blot. It is important to note that the levels of double-stranded RNA will vary per construct, as seen in the middle column using the current IVT method. We can see differences in the signal between EGFP, FLuc, beta-gal, and Cas9 mRNAs. On the right, we can see that using CleanScript IVT, all 4 samples result in lower double-stranded RNA signal relative to the legacy IVT method. This is also summarized by semi-quantitative values in the table on the left. This phenomenon of decreased double-stranded RNA production in the CleanScript IVT method is reproducible across various reaction scales. Here we have added 2 micrograms of mRNA from a 20 ml IVT reaction, showing similar levels as the 3 ml IVT CleanScript reaction by J2 dot plot. And finally, for good measure, because this assay, like most immunoblots, can introduce noise in your measurements, we have a full technical replicate panel on the right, again showing similar reduction levels between current and CleanScript IVT. Now that the enzymatic reaction using CleanCap Cap1 analog has been optimized for higher yields and lower byproducts, we shift our focus to the downstream purification processes. mRNA purification is required to remove salts, free NTPs, unincorporated cap analogs, and all proteins used in the synthesis and quenching reactions. We also need to remove our starting DNA template. An ideal purification process will go one step further and remove any truncated or degraded mRNA fragments and further reduce double-stranded RNA contaminants. Some of the more common purification processes are listed here. There are pros and cons to each method. The first 2 options, lithium chloride precipitation and silica membrane spin column, are not scalable as they both rely on centrifugation or well-controlled vacuum pressure. Of the next 3 options, cation exchange chromatography is a scalable process that will purify the mRNA, but is not capable of the extra ideal polishing steps, such as removing truncated mRNA products or decreasing double-stranded RNA. Reverse phase HPLC is known to deplete mRNA of double-stranded RNA contamination. However, this often comes at a great loss, with the average recovery from this HPLC method at only 50% of your starting material. Thanks to the advances with CleanScript IVT to help reduce double-stranded RNA forming in the first place, we will now focus on removal of truncated products by oligo-DT affinity chromatography. Here I am showing capillary electrophoresis traces from a fragment analyzer, pre and post from left to right, oligo-DT chromatography. In parentheses, we report a purity measurement using the software's integration or smear analysis. As you can see, oligo-DT chromatography results in mRNA quality improvement. On an EGFP construct, the quality goes from 72 to 77% purity as any untailed molecules are washed away. In an FLuc construct, quality can improve from 56 to 72%. In beta-gal, similar improvements can be achieved from 57 to 72%, with significant reduction in the front left shoulder of your main peak. Additionally, oligo-DT chromatography of a Cas9 molecule of length of a 4.5 kilobase length can improve from 54 to 72% purity. In summary, this affinity chromatography allows for the selection of full-length mRNA products by the poly-A tail. This is a scalable and universal mRNA purification process that removes degraded or truncated sequences. It also serves as a secondary clearance of residual proteins and DNA in data not shown today. To summarize, we have taken our already streamlined mRNA manufacturing process using CleanCap technology and further improved it for high yields with low double-stranded RNA contaminant for an even more robust and scalable workflow. With CleanScript IVT method, we can produce twice as much Cap1 mRNA with half the amount of double-stranded RNA byproduct in 5 simple process steps. Our process steps are not showing up. In brief, we have the sequential one-pot IVT, DNase, and Prot-K reaction, all of which serves to transcribe your mRNA, digest your DNA and proteins to small amino acid or oligo fragments for easy clearance through a size selection in a TFF or tangential flow filtration. mRNA is then polished by oligo-DT chromatography and can then be buffer exchanged into your final formulation before 0.2 micron sterile filtration. To end, I want to take you back to our case study and share results from improved IVT processes. Again, our study design looks to compare N1-methyl pseudo-U modified F-Luc mRNA, both using our CleanCap AG Cap1 analog, looking at differences between the legacy IVT and the new CleanScript IVT method. All mRNAs were produced at TriLink, analyzed before formulation into LNP, and then delivered into CD1 mice by tail vein injection. Again, we measure for our key mRNA quality attributes to assess the final product before in vivo experimentation. As you can see, CleanScript IVT produces 8.7 mg per ml in this experiment, which is significantly more than the legacy IVT method, resulting in 4.7 mg per ml from this batch. The mRNA integrity, as reported here from IP-RP-HPLC analysis, shows extremely high quality in both F-Luc samples thanks to the oligo-DT polishing step. Capping efficiency is maintained at 97 to 98% using either IVT protocol, which is important for our comparison of mRNA expression in vivo. Double-stranded RNA contaminant by J2 dot blot assay shows a decrease when using CleanScript from a relative 6.6 nanogram per microgram in legacy IVT down to 2.8 nanogram per microgram. And finally, the clearance of our residual DNA and proteins are similar for both mRNA. By now, I have shown that the CleanScript IVT with CleanCap analog improves current manufacturing processes with more mRNA volume per IVT and less double-stranded RNA byproduct. We next compare the mRNA expression in vivo, again using whole body luminescent imaging. Results show one representative animal from each cohort of 5 across all 7 imaging time points, again from left to right, starting with 3 hours to 96 hours post-mRNA delivery. The top panel shows the representative CleanCap legacy IVT test subject, and the bottom panel shows a representative CleanScript IVT animal. As you can see, luminescent signal levels from CleanScript are higher than CleanCap based on the intensity scale shown at the left. By plotting all data points from each cohort of 5 animals as a mean flux with standard deviation over time, we can see that CleanScript in purple reaches the highest signal intensity of mRNA expression at 6 hours and maintains similar expression levels out to 9 hours before dropping down at 12 hours. As previously seen, the CleanCap mRNA expression peaks at 6 hours post-mRNA delivery, but drops steadily for the next 6 hours. By comparing the integrated area under each curve, we find that optimized IVT conditions result in significantly higher mRNA expression in vivo. We next compared cytokine levels from the two blood draws as a means of comparing overall immune response to the different mRNA batches. In general, we observed that cytokine levels in response to CleanScript, as shown in purple, are lower than those observed in response to the legacy IVT, shown in blue. Taken all together, our mouse study data shows that all LNP mRNA delivery does result in mild immune responses, again, as to be expected. But the CleanScript IVT elicits less of this non-specific immune response while producing more protein expression relative to the legacy IVT condition. I'd like to again correlate our in vivo results back to our in vitro results. This study compared two Cap1 F-Luke mRNAs with varying levels of double-stranded RNA contaminant, a known trigger for pattern recognition receptor signaling pathways. In summary, today I have shown the benefits of using CleanCap Cap1 analog for a streamlined mRNA manufacturing process. CleanCap mRNA reduces innate immune responses compared to other CAP analogs and significantly increases mRNA expression in vivo. This is done at a reduced cost and quick turnaround time by the CAP analog co-transcriptional IVT. In addition, improvements to the mRNA synthesis process using our new CleanScript IVT method show further improvements in mRNA expression in vivo. CleanScript IVT allows for increased mRNA yield with less immunogenic double-stranded RNA byproduct. And finally, when considering your mRNA needs, let's refer to this summary table for manufacturability and function. For the highest IVT yields, a co-transcriptional CleanScript IVT using CleanCap1 analog results in 8 to 10 mg per mil in only 5 processing steps. mRNA integrity can be improved by oligo-DT affinity chromatography, and capping efficiency by the CleanCap and CleanScript methods results in 95 to 99% Cap1 capping efficiency. With no additional purification, CleanScript results in the lowest relative double-stranded RNA byproduct contaminant. And from data in our mouse study, we found that in vivo expression was highest when using the CleanScript CleanCap Cap1 mRNA process. I'd like to end today by highlighting the use of CleanCap in COVID-19 vaccines. While there are several research programs ongoing, shown here are the research articles using CleanCap in the Pfizer-BioNTech vaccine, which is currently in use, as well as a self-amplifying RNA vaccine approach by Imperial College London, taking advantage of the flexibility of our CleanCap offerings, utilizing the CleanCap AU analog. TriLink is headquartered in San Diego, California, where we recently moved into a world-class facility containing 15 ISO Class 7 and ISO Class 8 engineered GMP customer suites. We offer both catalog and custom small molecules, including oligonucleotides and highly modified nucleotide and triphosphate and with custom chemistry. As well, we can start all the way from your plasmid DNA production needs through mRNA production using our CleanCap technology. Everything can be done in a GMP compliant suite. And we also offer analytical services looking at key quality attributes of your final product. More specifically, for custom mRNA manufacturing, we can walk you through the entire process, again, starting from your plasmid design, your capping strategy, modified NTPs, and custom mRNA synthesis. Our capabilities include catalog and custom-made long RNA and mRNA, anywhere from 100 to 12,000 bases. Customizations can include your scale, modified nucleotides, capping, as well as post-in vitro modifications if necessary. With that, I'd like to thank and acknowledge my team at TriLink Biotechnologies. This study and technology would not be possible without the help of our entire research and process development team. Additionally, for our case study, we utilized Precision Nanosystems as our first CRO for LNP formulation and Charles River Laboratories to perform the in vivo mouse study. A special thanks to Mike Templin for his consultation in our study design. You can reach us at trilinkbiotech.com with any questions, concerns, or manufacturing needs. Thank you for your attention, and I'll open it up for questions.
Thank you, Jordana, for an excellent presentation today. I see we've got quite a few questions in already, but for everyone else, we'll give you some time to enter any questions in the Q&A box, which we'll try to address. Before we get to that, I'd like to thank BIA Separations, Arcturus Therapeutics, Cytiva, TriLink, and Intertech for sponsoring this digital week and making it possible. Also, be sure to check out the resources list to the right of your screen where you can download a few featured white papers. Now let's get back to Jordana to begin the Q&A. Jordana, as I said, we do have quite a number of questions in, so I'm going to try to read them out. Let's see if we can get through as many of these as possible in the time that we have left. First question I have is, does Cap1 capping make mRNA less immunogenic and consequently less susceptible to degradation?
Yeah, that's a really great question. It will always depend on the application or cell type or animal of choice for your mRNA studies. We do find that Cap1 elicits less of an immune response than Cap0. As well, in terms of susceptibility to degradation, none of the data shown today can actually address that directly. But we do know that other published in vitro data can show that Cap1 is actually decapped at a slower rate, which would suggest the message will last longer than a Cap0, which can decap at a quicker rate.
Okay, great. Next question. By what mechanism do you imagine does CleanCap reduce dsRNA formation in comparison to ARCA?
Wow, another really great question. My best theory to present to you today is I want to point out that the ARCA IVT reaction has a starvation of one of your building blocks, your GTP. And this was done to push the capping efficiency or initiation with the ARCA cap analog. But by reducing your GTP, your polymerase will run out of that substrate, essentially halting what would be a very processive transcription reaction. And what you're left with is an excess of your ATP, UTP, and CTP, which potentially could drive the aberrant T7 activity, ultimately resulting in double-stranded RNA formation.
Okay, perfect. Next question. How does CleanCap compare to enzymatic capping?
Yeah, another really great question. I know that wasn't shown today in our case study, but I would point you to the current literature comparing the Moderna to Pfizer-BioNTech vaccine. Both capping strategies are efficient in terms of mRNA function. It's what we wanted to focus on today was the cost savings and streamlining of the overall process.
Okay. Next question. How does CleanScript IVT perform with larger constructs of greater than 9 kb?
Yes, we showed the results up to 4.5 kb with our new CleanScript IVT method. We're currently still working on the development of the 2.0 version of the method that will address longer constructs. As you could imagine, having a higher reaction yield in a fixed volume will create some solubility issues. So it's something that we're still currently working on.
Great. Are you going to sell CleanScript IVT as a kit?
That one is a little above my pay grade in research and development. So I would defer you to our marketing and sales team. Feel free to reach out to them on our website.
Okay. Next one. What method is used to measure percent integrity of mRNA?
Today I actually showed two different methods. A fragment analyzer, capillary electrophoresis, and we use the software's integration protocol to perform a smear analysis. We also have HPLC methods with a UV detection, again, where we can integrate the area under the curve of our main peak and report a percent purity in that way.
Perfect. I'm going to go a minute or two over just to get these last two questions in. Next question, is high yield of RNA with CleanScript a problem in terms of solubility?
Yes, great question. So I alluded to that in answering a previous question. I'm not at liberty to divulge a maximum yield at this time, but the reaction at the end of your IVT is very viscous. So this is why we're still working on developing a process for longer mRNA.
Got it. And the last question I'll ask is, does the oligo DT purification remove the dsRNA too?
Great question. So I didn't show data supporting that today. Well, it can remove some level of the contaminant. It is not our preferred method, as the reduction is not as significant as other options we have in our toolkit.
Okay, perfect. Well, it looks like that's all the time we have for questions today. I'd like to thank you, Jordana, again for a great session. I'd like to remind everyone that this session was recorded. You'll receive a notification in 24 hours when the on-demand session is available for viewing. And before you log off, I'd like to encourage you to please take a moment to fill out the feedback form so we can continue to improve your Digital Week experience. On behalf of Informa Connect Life Sciences, thank you for attending and hope you all have a great day.